So let's get into it. Let me just grab a quick sip. So inquiry question is, how does mutation introduce new alleles into the population? So module 6 is a really nice segue from module 5, where now when we look at mutations, firstly we're going back to our processes of replication, translation, transcription, and talking about mutations affecting those processes. Okay, here we are. So first question we have here is that we're going to look at is, how do mutagens operate, the different types of mutations. We're going to talk about somatic versus germline mutation, and then talk about the effects of mutations on coding versus non-coding DNA, and then look at causes of genetic variation, and finish off by talking about population genetics. So we're going to talk about mutations, gene flow, and genetic drift. So here we are. Inquiry question is, how does mutation introduce new alleles into a population? Remember, a mutation, firstly, is a permanent alteration to the nucleotide sequence of an organism's genome. This is where we are permanently altering someone's or rather natural circumstances or their own system leads to the alteration of someone's own nucleotide sequences, which then changes the proteins that they are able to make. So we'll firstly begin by looking at the mutagens. What are the things that actually cause these mutations to occur? And we're going to talk about electromagnetic radiation first of all. Electromagnetic radiation, as the name suggests, refers to the waves that are emitted by electrically charged particles. They interact and ionize atoms that they encounter, and there is a danger, and the danger of radiation lies in its penetrative ability. So electromagnetic radiations can penetrate quite deeply, and they can travel into the nucleus, and they can adhere with DNA molecules, and they can lead to the creation of free radicals. So it's not very good at all. And how does it actually cause mutation? So once it actually penetrates in, there's a lot of energy, right? Electromagnetic radiation, there's a lot of energy input, which actually may alter the way that the DNA is bonded, and it can change the chemical composition of the DNA as well. It can also lead to the rearrangement of the structure. So here we are. There are examples. For example, ultraviolet light is an example of electromagnetic radiation, and it is radiation from the sun which, if encountered in high doses, in daily high doses, can be harmful. However, we do encounter it daily, and that's why we know that there is huge stress on the importance of wearing sunscreen, being sun safe, wearing protective clothing, because we know that there is an increased risk of melanoma from it, which is skin cancer. So UV light is harmful in high doses, and what it does is that the incoming photons actually excite the adjacent pyrimidines. So these adjacent pyrimidines, which includes T and C, so thymine and cytosine, so it incites them, and it then causes them to actually turn around and bond to each other through covalent bonds. So instead of the hydrogen bonding here, that hydrogen bonding is broken so that the C and the T can bond to one another. And on an average summer's day in Sydney, a person will acquire around 20,000 pyrimidine dimers. So it does happen. It is a very, very common thing. But, and there are lots of mutations that occur, but the thing here is that we do have our own repair systems that do repair some of these changes. So we'll talk about what happens if those changes aren't repaired. But this is the first mutagen that we look at, electromagnetic radiation. Now let's have a look at chemicals. Chemicals are also mutagens. We have radioactive agents, so agents like uranium, and they can release radiation, including alpha and beta particles, and they are going to be high energy particles, and they are going to they can include alpha and beta particles, which are high energy, and they can also include gamma waves, which is actually a form of electromagnetic radiation. And both of them can then, or all three of them, can then penetrate the cell and interact with the DNA bonds, again creating structural disruptions. In terms of metals, so the metals that are mutagens include arsenic, nickel, and cobalt. And so being exposed to them for a long amount of time, it can affect the integrity of the DNA repair system. So we have our own inherent repair system that essentially stops the proteins from, that basically fixes any mutations or tries to fix as many mutations as possible. But if that repair system is affected, which is the case with metals, that they can affect the DNA repair system, they can actually stop proteins from being able to recognize base pair mismatching. So it stops that recognition of base pair matching. So any incorrect base pair matching, it stops that from being detected. And so as a result, we have as a result, that's overlooked and that mutation goes unnoticed. And it reduces the fidelity of the DNA replication, so more errors occur. And nickel especially inhibits the ability for histones to condense DNA. So histones are really important because that's where they're wrapped around DNA and they essentially protect our DNA. And if they essentially, and if nickel affects the ability for histones to condense that DNA to get that DNA to be protected, it can then lead to the chromosomes being in a bad formation, and that can of course affect the protection of the chromosomes. Last one, naturally occurring mutagens. So naturally occurring mutagens includes viruses. So these are things that are out there in nature that can lead to mutations. So viruses are an example of this, and what viruses do is that viruses replicate by inserting their own genetic material into the host cell. So they insert their own genetic material into the host cell and they disrupt the normal cell function. And by doing that, that can lead to lasting changes to an organism's genome. So viruses don't have the machinery to actually make their own proteins. So every time a virus, so they need basically another body to survive. Like they need a human body to survive. So they get in there, they're going to get into our cells, gain entry, hijack our whole transcription translation machinery to then put in their own genetic material so that that material can be produced, so that their proteins can be produced. And these are some examples of viruses. So HIV, of course, which then leads to AIDS. Hepatitis B, Ebola virus is another one, adenovirus, influenza, and bacteriophage. And bacterial infections, which is bacteriophage right there, what they do is that they induce inflammation. So they cause inflammation, and that reduces the efficiency of our DNA repair systems. And it also then leads to the increase in mutations. And a good example of this is, for example, Helicobacter pylori, which is a bacteria, and it has actually been linked to the development of stomach cancer. Remember, cancer is that unhindered replication of the cells, right? And what happens is, if our DNA system is repair system is unable to repair that mutation, the cells keep replicating, and then that then leads to cancer. And in this case, there has been correlation between Helicobacter pylori and the development of stomach cancer. So now we know what these mutagens are. Let's talk about the effects of these mutagens.